Good morning, everyone. Okay, so Adriaan van Niekerk from Stellenbosch University, Department of Geography and Environmental Studies. And you can see the title there of this specific study. And you probably know this plum marbling problem better than I would, caused by the plum viroid 1. And as you know, the symptoms are irregular skin discoloration, small hard fruit, etc. And these symptoms often only develop when the trees are grown or when they bear fruit, which is often too late. And there is a detection methodology developed by my colleagues, colleagues at the university. Oh goodness, please start that again. Just want to get this thing going. It's not working. Okay, at the bottom there you can see Bester, Milan, and Maria. Did a research on this, and they established a little orchard of 10 varieties of plums, and inoculated half of the orchard. And the question came up, and they developed this PCR test to test whether these orchards are infected or not. And although this PCR test was a huge improvement, it's the first way of detecting this viroid, Hoortcrow approached me to see if there's a possibility of identifying this viroid remotely, meaning without contact with the actual tree. Ideally, to eventually be able to remotely, either via drone or even satellite, detect these trees that are infected by this viroid. The first step, however, is a proof of concept just to see if this viroid can be detected using spectroscopy, and spectroscopy is simply using different wavelengths of light, the reflection of the light from the leaves of the trees, to see if we can differentiate leaves that are affected and those that are not. So if we can do that, we can build a sensor that would be able to either be used in the orchard or remotely. All right, so these are the leaves that were picked from the experimental orchard, 10 varieties, and it's more or less also the layout of the orchard, where two rows, so the columns are the different varieties, and the rows are either affected or inoculated or not, the red ones. And there were 60 leaves, which isn't a lot of information. The whole idea with this study was to see if AI can be used to differentiate also between these infected or inoculated leaves. So because there were only 60 leaves, 30 that were inoculated, or 30 that were harvested from trees that were inoculated, and 30 that were not harvested from trees, or harvested from trees that were not inoculated, we segmented all of these leaves into smaller pieces, not only to increase the number of samples, but also, as you can see on this photograph, or the scan, that there are a lot of variation in light as it is reflected from the leaves. You have white spots, which we call specular reflection, and then you have areas that are more in shadow or less illuminated, and that causes variation. And that is a challenge when it comes to machine learning, but also, as you can imagine, if we want to use the same methodology in the orchard, you need actually a lot of variation, because although this was taken in a lab situation, if you want to develop something that will work in the orchard, you actually need to make provision for these variations. So by segmenting these leaves, we could extract thousands of different samples of the spectral properties of each of those little blocks. And what you see on the graph, the top left, is, well, the whole graph at the top represents the wavelengths of that the scanner provided. There were two scanners we used, one in the visible range, the range that we can see, and that is indicated in the colors there. And you can see that there is, maybe you cannot see, but it's a slight difference in the reflectance in the green band. And actually what we found, well, this is showing that leaves that were inoculated are slightly greener than leaves that were not inoculated. But the difference is very small. But as the wavelengths increase, the difference becomes larger. And this is the average signal from many samples, and there's a lot of variation between them. But as you can see towards the longer wavelengths in the shortwave infrared, the difference becomes larger. Now, in machine learning, what you would normally do is you would take those samples and you would split randomly the samples into a training set of 80% of the samples, and 20% you will leave out for testing the accuracy of the model that you produced. And that was the first experiment that I carried out. And the results were very strange. The accuracy was 95%. I couldn't believe it because those leaves look exactly the same to me. So I said, no, this must be a problem with the experiment. I'm going to redo it, and this time I'm not going to do a random sampling. I'm going to sample one leaf per image bed and use all of the corresponding segments for the testing the model. So one of all of those leaves were taken. Oh my goodness, I don't know what I'm doing. Going forward and backwards. Okay, I see there's actually a pointer to the wrong, I was using the wrong pointer. Okay, now I got it. So there you can see one of those leaves per bed was used for testing, and the others were used for training. And then the next experiment was to use one column for testing and the others for training. And then use first the east column and then the right column. So I tried everything. I threw everything at this. And the results all showed accuracies of more than 90%, 95%. This was astounding. I've never seen anything like this. And then the next part of my question was, okay, this is for the full spectrum. So that, having a sensor that covers the full spectrum is expensive. What if we build a sensor that only can sense in a few wavelengths? That will be much cheaper. So I took out wavelengths, and the top right graph shows that if I use 12 wavelengths, then I can get a 95% accuracy. Down to 4 brings to still 95, and then if we only use 2 wavelengths, it's 94%. So with much fewer data or a much cheaper sensor, we could still get 94% accuracy. Okay, I'm almost done. The so what is, yes, we can detect, but this was done in a controlled environment, just a proof of concept. The next step is to expand it with more data and mature trees. Thank you very much.